Yep, thank you, Peter. I'll just hopefully bring up my slides. Can you see them?
Yes.
Okay, and I'm also just going to kick off now. You might hear me press the key, a little process in the background that I will come back to and explain a bit more about. So yeah, Peter, you'll give me a 5-minute warning, is that right?
Yes.
Okay, so a bit of a gear change now. So I'm going to talk about predicting antimicrobial resistance to tuberculosis, essentially. And I've talked a little bit about this before, but crucially, since I last spoke, we've set up a global cloud-based platform. So that although this talk is going to be about TB, that it does involve a pretty heavy diversion through SARS-CoV-2, but I'll try and walk you through it. Okay, so we're a pretty small group based in the John Radcliffe Hospital in Oxford. Charlotte Lynch, who's my postdoc, is also at the meeting, and we're part of a much larger unit called the Modernising Medical Microbiology Group. That's the group of people at the top, also in the hospital, and that's a very diverse selection of infectious disease clinicians, statisticians, software engineers, wet lab people, bioinformaticians, and so on. And broadly, one of the things that we're all working together towards is sort of shifting the paradigm in clinical microbiology from culture, which is shown here, where you take a sample from a patient, you grow it on an appropriate media, which can often be quite archaic, often on an agar, on a petri dish, and then you literally just test which antibiotics work or not using bits of filter paper or something, and instead just saying, well, actually take that sample, putting it into a whole genome sequencer, get a lot of reads out, process those, get the consensus genome, look up the mutations in some kind of database, and from that infer which drugs you can treat the patient with and which will not work. And what we focus on in particular, because those methods are inferential, there are times when they come across mutations that are not in that catalogue, and so we need predictive methods that work de novo. And there's pretty big gains to be made in shifting to genetics. So for example, with TB, that left-hand loop can take up to about 2 months. So how are we doing this prediction? We've sort of ended up in a slightly haphazard way with this 2-step process. So we use machine learning to rule out mutations that are likely to have no effect. So in other words, the infection will be susceptible. And then we use much more heavyweight relative binding free energy calculations to look at the remaining mutations in more detail. Now, what's kind of was obviously very interesting, but has been taking up a lot of our time is the COVID pandemic came along, and partway through that, Oracle started to talk to the University of Oxford, and just over a year ago, so last May, it was agreed that Oracle would donate or give Oxford free use of its cloud infrastructure for 10 years to do pathogen sequencing processing. And so as a result of that, we've been working flat out to stand up a platform focusing initially on SARS-CoV-2, which we've called GPAS. And what I did at the start of the talk was I uploaded from my computer here. I'll just check how many it was. I think it was 15. Yeah, 15 COVID samples that have gone through an Oxford nanopore sequencer. So actually, I'll skip that, I think, in the interest of time. That was just a little screencast. And actually, what GPAS is quite trying to do is it's just trying to do what's the kind of quite boring processing step in the genetics. So what you get out of the genetic sequences, I'm not sure how familiar everyone here is with it, but you get basically a big collection of reads that may or may not have come from your organism of interest. So from a human sputum sample, for example, you may not have, but you may have some Mycobacterium tuberculosis. You're going to have some other bacterial species that are also present in the upper respiratory tract, and you're also going to have a lot of human DNA as well. So what you have to do with COVID, for example, is you have to first of all remove all the human DNA, because you can't upload that to the cloud, because that's personal information under GDPR regulations. And then you have to essentially work out which reads are COVID and which aren't. And then when you've got the reads that are COVID, you have to use those to construct or infer what your consensus genome is, which is a bit like a sort of jigsaw puzzle where you haven't got a picture of the box on the box. And anyway, it's one-dimensional. Now, this isn't HPC. So a single SARS-CoV-2 sample only requires about half a CPU hour to process in the cloud, but we do have to do it at scale, and we do have to do it reliably and reproducibly. So there are over 3 million SARS-CoV-2 raw genetic files deposited in the ENA, for example. So in the cloud, we have and this is typical for bioinformatics, we have quite a complicated workflow that's described using Nextflow, which is a workflow language that's used quite widely in bioinformatics. And that in turn, the jobs are run within Docker containers that's orchestrated by Kubernetes. So very quickly where we're up to. So the idea is GPAS will be free to low and middle income countries. And we've actually set up a spin out that's acting as a not-for-profit so that we can engage in tenders with public health agencies in high income countries. We've had to set it up very carefully so that organizations own their own data. No one wants to be the next South Africa, for example, with COVID. And they then are able to choose how they share their data. So they can choose to share samples with what we've called the relatedness service. I'll talk a bit more about that in a minute. Or they can actually choose to grant access for specific samples to other organizations. Now, at its core, GPAS is just processing samples. And in some sense, if you view, what was perhaps originally called grid computing, maybe now cloud computing, as a commoditization of computational resources, if you unpack that in different ways, you could also view something like GPAS as trying to commoditize that processing step that everyone has to do when they do some genetic sequencing. And one of the main advantages, probably actually the USP for GPAS, is not necessarily the speed or the reliability, but by moving everything into the cloud and then giving people control over whether they share their samples or not, is that we can start to do some very, very interesting epidemiological things. So for example, if enough countries share their samples, we can detect importation events. So what you would see is you would see a sample that is unlike any other sample you've previously uploaded, but it would be very similar. So only a couple of SNPs away, or identical even, to samples from a neighboring country. But it also lets us potentially detect novel variants, because those would be samples that not only you haven't seen anything like, but no one else has either. Of course, though, COVID is on the wane in high-income countries and the Northern Hemisphere at least. But even in the UK, as sort of the latest Omicron flavors, BA.4 and .5 are on the rise. We also see quite a lot of recombinants. So watch out for Deltacron, which, although most of our recombinants have been Omicron recombinants, I think Deltacron always makes me think it's some kind of transformer or something. But so we're pivoting back to what we know best, which is TB. Now, a lot of TB's unique features arise from its very, very slow doubling time. And around 10 million people develop TB each year, and around 1.5 million people die annually. And before the pandemic, TB could reliably kill more people every year than any other infectious disease. So it kills more people than HIV and malaria combined. And since 2016, we've been leading a large international consortium that's collected over 20,000 TB samples from 11 countries. Each was whole genome sequenced and tested against 13 different antituberculars. And as a result, we were invited to work with the WHO to produce their first catalogue of mutations in Mycobacterium tuberculosis. That was released, I think, exactly a year ago, so about the 20th of June last year. So we're working now quite frantically to put TB into GPAS to hopefully enable a fairly cheap and rapid way of coming up with individualized treatment plans to treat TB around the world. It's worth thinking a little bit about the differences between SARS-CoV-2 and TB. The point I'm drawing out here is that SARS-CoV-2, you don't really take any decisions, at least not yet, at the patient level. It's more at the public health level. So it's how many, what's my mix of lineages? Have I got anything new arising? How many cases do I have? That sort of thing. Whereas for TB, it's very different. So here you absolutely care about the individual patient, because what you're trying to get to is an antibiogram. So that's a list of antibiotics and whether or not you can successfully use those to treat the patient. Now, TB is a bit unusual in that you have to treat it with combinations of antibiotics. So, for example, shown here are the 4 antibiotics you'd be given if you had normal susceptible TB. And you'd also have to take those for about 6 months. So it's a difficult disease to treat. Now, going back to the catalogue, what we often find is, because there's quite a long tail of rare and novel mutations in the genes, is that even when we have a lot of entries in a catalogue for a particular gene, we will often encounter mutations that just haven't been seen before. And in that case, the method, because it's inferential, is forced to return an unknown result, as it's done here for rifampicin. So here, clinically, we're not sure whether we can treat this patient with the standard regimen or whether we have to move them onto an MDR regimen, which is usually not only a lot longer, more expensive, but actually quite unpleasant from a side effects point of view. So what can we do for prediction? As I said, we're taking 2 approaches really, the first of which is machine learning, and then the second is some relative binding free energies. So this first bit is work that's been done by, first of all, a project student, Dylan Adlard, and then has been taken on by Charlotte Lynch. So what is rifampicin? It's quite an unusual ring-like molecule, and it binds to the RNA polymerase in TB, which is itself a pretty big protein complex. Fortunately, we have a structure for TB, which is shown here. And what it does is it binds in such a way to prevent the extension of the RNA. So for machine learning, what we've done is we've taken all the 75,000 clinical samples that Cryptic has either collected itself or has kind of scraped from around the web. And from that, we then filter down to individual samples where there's only a single mutation, single non-synonymous mutation in that gene, so we can infer that it's having the effect. And then we add our labels, which in this case are the results of phenotypic tests, so whether it's resistant or not. Then we add a whole slew of protein features, so structural, thermodynamic, biochemical, evolutionary features. We do a test-train split, as you might expect, and then we train a variety of machine learning models using crossfold validation. Now, we are very much at the minute focusing on pretty simple models, and we're putting all of our effort really into the feature engineering, partly because as you might imagine, if we're to offer this through GPAS, ideally we'll need to explain at the same time why the model has made the prediction it has. So there's a sort of a tension between explainability and potentially performance as well.
Phil,
Yeah.
5 more minutes.
Okay, thanks, Peter. So Charlotte's been spending a lot of time recently. We've written a Python package to make this easier called SBML Core, just helps us build the feature sets more easily. And here are some distances, for example. Now, just a very, very simple model like logistic regression does pretty well, but it's pretty hard because we've got a very imbalanced data set. So we've got relatively few unique mutations that confer resistance, which makes life a little hard. Something like a random forest does a bit better, as you might expect, because it can add some kind of non-linearities. And looking at the feature importance plots, a predictor of protein stability does pretty well, as does a distance, which is sort of what you'd expect. So that's machine learning. It's a very active area of development for us. We've got work here. We've got work on the fluoroquinolones, also on isoniazid and pyrazinamide. So more soon on all that. I have shared some relative binding free energy calculation results before, but I think it was on a much smaller protein, DHFR. And here we're going to look at the RNA polymerase, and these are the 6 mutations we're going to study, as shown there. And then also PhD student of mine, Alice Brankin, has tried a very similar approach on the DNA gyrase, which is where the fluoroquinolones bind and where you see resistance, as shown here. So it's still, I mean, at nearly 200 kilodaltons, it's a big old protein. So missing out an awful lot of methods and an awful lot of, I guess, numbers, simulations we had to do, but we needed 2 separate compute allocations on Archer to do these calculations. And what we found was that for rifampicin, I should say the dashed vertical line is the clinical threshold above which we have to, the delta delta G has to lie for us to classify this as resistant. And we can see that all 3 of the red bars are indeed above that line, so they would be classified as resistant. And it's also interesting to note that we don't really care about quantitative accuracy a bit, which does throw reviewers of our papers sometimes. We just want to know which side of that dashed line does it lie, because that's the clinical paradigm. However, for the 4 mutations that have no effect, 3 of them, the confidence limits straddle the clinical threshold. So we end up returning we basically say, I don't know, I can't tell. And only 1 of them can we definitively say that it has no effect. So that's a bit of a mixed picture. And for the DNA gyrase, the situation's a little bit worse. Primarily, we think it's a mixture of the mutations are bigger perturbations and more kind of harder to deal with. So D94G involves a charge change, which is always difficult. But also the size of the signal, so the size of the shift in delta delta G is smaller for the fluoroquinolones than it is for rifampicin, which makes it difficult when your confidence intervals are this big. So I will stop there. Bit of a whistle-stop tour, bit of a story as well. And I've actually realized I haven't shown you, maybe if I've got a minute. So very early on, when Peter was talking, I uploaded those 15 nanopore samples to GPAS. This is the web portal. It's these ones here. You see the ones below. I was practicing slightly earlier today. And if we go in and look, we can see that we've found out all the lineages of each of these. And these would have taken about 10 minutes to run in the cloud. And crucially, we can see we've got a mixture of BA2, BA3. These are all Omicrons. This is a Delta. That's an Alpha. And I've all made these up just to kind of make it interesting. And if we go in and look at a particular sample, we can see where other similar samples have been uploaded previously in the system. And then also, in this case, there's been one other sample uploaded that's identical, which was probably the one I uploaded before this one. So shortly, hopefully, certainly by the end of the year, we'll be able to do this for TB as well. And then the plan will be to start offering those machine learning models kind of on the back of the antibiograms, so that there is information there for the clinicians when the catalogue essentially is stuck and doesn't know what to do. All right, I'll stop there and happy to take any questions.